Hello, my name is Ben Lahner. I'm a PhD student at MIT in the Computer Science and AI lab, and I'm advised by Odaliva. I want to say thank you to Voxel51 and Jimmy and Jacob for inviting me to talk today about using computer vision to understand biological vision. So first, just briefly a little bit about me. As I was born and raised in Minnesota. We got two seasons, summer and snow, and then I went on to go to school in Boston, where I did my undergrad on the right side of this picture at Boston University. Then I moved over to the left side of the picture across the river to MIT. And one thing that I like to do with my family, my brother, and my dad is we like to take hiking trips out west. So we went to Glacier National Park. This photo looks like it was taken at night, but it was during a solar eclipse at high noon in Wyoming. Renting llamas to go hiking in Colorado or New Mexico. But tying this back into the presentation here, on our hiking trip at Glacier National Park, me, my dad, and brother were hiking down the trail, and the trail emerged into a clearing. And we also at the same time, about 10 feet away from us, there was this big grizzly bear. And oddly enough, we all had different reactions to it. So my brother froze, my dad wanted to run away, and I whipped out my phone to try to take a picture. And thankfully, my brother and dad were there to kind of shove me along, keep going, don't disturb the bear. Eventually, I managed to get one little snapshot of it. But this represents a good introduction to the goal of computational neuroscience, where we have some information about the world, some visual information, in this case, a bear, and elicits some behavior, whether it's just recognizing it's a bear, whether this information deserves attention, memory, whether to fight or flight, or take a picture. And this is all mediated by our nervous system, in this case, the brain. So the question becomes, well, what's going on inside the brain? And so the goal of computational neuroscience is to find a mechanistic explanation of how the nervous system processes information to support cognitive function and behavior. So it looks like all the pieces of the puzzle are laid out in front of us. Shouldn't be too hard. We know the inputs, can measure the model, know the outputs. It's actually more complicated than that. First of all, the brain is notoriously very difficult to get information from. This is where it's located in your brain, just for reference, in relation to your head. But when a neuron fires, we can measure a couple signals from it. First of all, is the electrical field it emanates, and we can use an EEG cap to collect that signal. And orthogonally, it has a magnetic field that we can collect with MEG. And also, when a neuron fires, there's blood flow to that part of the brain, and we can use fMRI to measure that blood flow. Or we can even open up the brain and put and measure the electric field directly on top of it with electrocorticography. And there's pros and cons to each, but the gist of it is they're all noisy, they're all expensive, and there's no catch-all solution. Ideally, we'd have something that can measure the brain and with really high spatial resolution, really high temporal resolution, but that just simply doesn't exist. So we have to make do with what we have. So briefly about the human visual system, is there's two dominating pathways. There's like the ventral pathway, that's known as like the what pathway. You think of this as like object recognition. And there's the dorsal pathway, that's more where. So it's like spatial navigation, some motion selective regions too. And so as you see like a visual input, first it enters cortex around the V1, V2 area in the back of the brain. And the neurons are kind of oriented to pick out some tuning properties, the spatial orientation, so you can get that, add some texture, and then goes through the brain, kind of put those together to get a shape, add some color. And finally, we have something that looks like a bear that some regions are able to decode far down the pathway, say that this is indeed a bear. And what I just described is decades of neuroscience research and modeling efforts to be able to come to that knowledge. So what I want to talk about today is the rise of DNNs as vision models, because models are really the core, they're the glue of computational neuroscience, links everything together. And then I'm going to talk a little bit about my work with spatial-temporal mapping of the brain and video understanding. And then lastly, touch on what's the future of DNNs in computational neuroscience. First, the rise of DNNs as models of the visual system. Before we had these deep neural networks, a lot of these modeling approaches were directly inspired by neuroscience. So you can imagine that if we know from neuroscience experiments specific tuning properties of V1 neurons, for example, that they do care about the orientation of the stimulus, we can build a model to then probe and predict neural activity. And so we can use pretty simple stimuli like this sinusoidal grading, try to map out like receptive fields of this region. But we want to go beyond V1 and V2. We want to try to get at regions further down the hierarchy. And so one region that is motion selective that V1 projects to strongly is this region called MT. And this is a similar like plaid black and white stimulus that's moving a little bit. And they used a model that kind of combines a V1 model with linear weights from MT model to try to predict that activity. We still want to go further. And so we're using more and more complex stimuli like textures and grayscale images of animals to try to probe different parts of the brain and try to model that response. And so here's some examples of some models that they're all inspired by neuroscience results, recognizing that the ventral visual stream is largely hierarchical, and you want to build these representations up from one another to be able to finally predict object recognition as one of the goals. But the problem is that these models are scientifically inaccurate, meaning that they're not robust, they're very sensitive to small perturbations in the stimulus. If you like translate it a bit, rotate it, you see a dramatic decrease in accuracy. They just didn't perform very well. But then around 2012, we had this breakthrough with DNNs. All of a sudden, we get this model that when you optimize it to perform the specific task, biologically meaningful task at that, like object recognition, we see that the internal representations explain IT, which is very far, which is like the end goal of, or the final region of visual perception. It explains that region very well and outperforms a lot of the control models like V1 and V2, like HMax and SIFT. And as you start to perturb the stimuli, this difference just increases dramatically. Performance gets better and better. And they also show that as the model gets more and more optimized on the task, gets better and better at explaining brain activity. And it's important to note that this didn't have to happen. This just kind of fell out of these DNNs. So this is pretty exciting because all of a sudden we have at our disposal a model that is image computable, it's biologically inspired, and it's very powerful predictors of ventral visual brain activity. And so how they're actually, some uses of them to model the visual system is using an encoding model framework, where you have some input, passes through a nonlinearity to arrive at some feature space, and then you do a linear projection onto the brain activity. And so all the interesting stuff, all the hypotheses happen in this nonlinearity right here. And so you can use that nonlinearity to test like the Bohr wavelets, some more simple sinusoidal models, could even be semantic models, or as we're talking about here, deep neural networks. So a study that I really like that uses this approach is by Murty et al. In 2021. And some background is that there are these regions in the brain that are thought to show very strong preferences specifically for faces, scenes, and bodies. So much so that people are saying that they are selective for them. people will push back saying, can't say they're selective because, again, collecting neuroimaging data is expensive. So really, you're just basing selectivity on a couple thousand, maybe 10,000 images that you've run through a scanner, with the space of natural images is immense. So there's no way you can say it's selective. And so they use this approach where they use DNNs to build a really, really strong predictor of neural activity. And then they did a high throughput test of tens of millions of images. It was like 3 million images to see if any like non-face image, for example, strongly activated the face network. And if it did, they would put that through the scanner to then test it on a real human. But what they found is that that wasn't the case. They really do seem to be selected for faces, bodies, and scenes, and then kind of switched it up. This use a GAN to generate maximally activating images for these regions. And again, these are unambiguously images of faces, bodies, and scenes in the bottom row. And another advantage with DNNs is that they are pretty interpretable, at least more interpretable than the brain. You can look at what they're paying attention to. And again, the network that they built, the DNN that they built for the face region is unambiguously paying attention to the faces, same with bodies and scenes. And so another way you can model brain activities using decoding models. You can think of this as mind reading. So it's very similar to encoding models. We have an input, goes through some nonlinearity, except in this case, we're kind of thinking of the nonlinearity. So it could be a semantic description, it could be motion direction, or it could be some behavioral readout like attention. And then use the brain activity to linearly decode back to that feature space. And so a recent fun example of this is doing image reconstruction with stable diffusion. You can see these red arrows right here is this linear model that is analogous to right here, the brain activity going back to the feature space. So linearly decoding brain activity from the back of the brain, again, that's going to be like your spatial orientations, like pure visual features. And then also, again, for these high-level regions, which are more semantic, these are the ones that are kind of pinpointing that this is indeed a bear. And they show reconstructing results using just visual features, and then just the semantic features, and then if you include both, and they show that including both gives you better reconstruction. And the third and last framework I'm going to talk about is representational similarity analysis. And the idea behind this is that when you see a bunch of images and you can measure your brain responses to that, doesn't matter how, but you measure your brain responses to that, you can compute how similar each image is to each other. And through computing this similarity or equivalently dissimilarity, you can populate this matrix on the right, this representational dissimilarity matrix, or RDM. And where each ij index is a pair of image I, image j. And this dissimilarity can be like 1 minus Pearson correlation, could be classifier accuracy, could be a couple of different things. But so the point is you populate this RDM. And the reason why that's important is because now we can start to take these usually non-congruent parts of computational neuroscience, like models, behavior, brain activity from different modalities and species, and put them in this common representational space where they are now directly comparable. So now we can start to do cool stuff with comparing brains with models and behavior. So a neat example of using this is establishing that DNNs are not only good models of the brain across space, but also across time, so using MEG and fMRI. So now touching on some of my work using building spatial-temporal maps and video understanding. There's this method called MEG fMRI fusion from our lab, where the principle behind it is your neural representation of a stimulus shouldn't depend on like where you're sitting. It doesn't matter if you're an MEG scanner or an MRI scanner, it should be the same. But the idea is that we can use these different neuroimaging techniques to take different snapshots of brain activity. So with MEG, you can get really good temporal representations. You can build this RDM across time. Now with an MRI, you can build these RDMs across space if you run the exact same experiment with the same subjects and the different scanner. And now these MEG and MRI, where previously they were not really comparable, now that they're in the same representational space, you can start to compare them. And you do, by correlating them, you can figure out which spatial regions are activated when in time start to build these high quality spatial temporal maps of neural processes. In this case, we're looking at vision. And just to note the time scale here is by 150 milliseconds, your brain has already kind of identified that this is a bear and is ready to make a behavioral decision about it. So it all happens like very fast. 150 milliseconds is eternity in brain time. So this is what it would look like in vision again. So onsets around 70 milliseconds and propagates up to the ventral and dorsal visual streams. And we can do the same thing with an auditory experiment where now they're listening to a bunch of sounds. And we can also build spatial temporal maps here. So we see the auditory system is on the side of the brain by the ears. So we can see the correlations start to light up around there and propagate throughout. Maybe not the left side of the brain is more tuned for human speech and stuff. So we can see that light up even more strongly in this panel. And we can use it to investigate other neural processes like visual memorability. So this is the work that I have in progress, ready to go out soon. And the idea behind memorability is that some images just stick in your mind better than others. It's just how our brains work. But memorability, it turns out, is actually this attribute of an image that's consistent across people, across time, where everyone's going to think that one image is more memorable than another. So what we did is we crafted the stimulus set with high memorable and low memorable pairs. So you'd semantically describe these all the same. It's like a baseball at bat or a jet skier on a wave. And these sets were controlled for other low-level features as well. But we can start to see where and when is the brain sensitive to image memorability. And turns out that we're just going to blow right past that 150 millisecond mark. And around 250, 275 milliseconds, that's when you start to see these significant correlations arise. And this is very late. And so this fMRI fusion is really good at pinpointing like hierarchies and activations that occur across space and time, since we really do need this really fine spatial and temporal resolution. And again, no single imaging modality can provide that yet. So looking at some of these results, again, we can see that memorability is distributed throughout cortex, both in the back of the brain and through the ventral stream. EVC is a good example here. This is early visual cortex, where the high memorable and low memorable curves are pretty much the exact same until around 250, 300 milliseconds, where the low memorable just stays at 0 while the high memorable just jumps right back up again. So now looping the brain activity, or sorry, the DNNs back into the game. We can ask, okay, what model is a really good predictor of both MRI or fMRI and MEG brain activity? And this is a pretty lofty goal. And so the idea is, well, this is too much for one single research group to do. So let's make a challenge out of it. So this kind of inspired and launched what we call the Algonauts project. And that was actually the subject of our first edition in 2019, by explaining the human visual brain. where we had 156 images, and we had MRI and MEG activity from each. And so the challenge, it's publicly released. We just invite a bunch of neuroscientists, computer scientists to compete and win a prize if they could come up with the best model that best predicts MRI or MEG activity. And so since then, we've done a 2021 edition. Instead of using images, we use short videos, 3 seconds long. We collected a very large data set of 1,102 short videos across 10 subjects in fMRI. I'll talk about more of this in a second. And then currently we have a 2023 edition that is live now. And we are using the natural scenes data set. If you have heard about it, it's a massive data set that was released, collected on COCO images with MRI. So again, the idea behind Algonauts project is to try to encourage interdisciplinary collaboration and educational resources. We provide tutorials and partner with the conference to then communicate the final results and have a nice day of talks. So in this edition, we're asking if you can build a winning encoding model. And again, we used the NSD experiment, which is from 8 subjects who all saw a large part of the COCO data set and collected via 7 Tesla scanner for really high quality MRI images. And they were kind enough to hold out a portion for the Algonauts 2023 challenge. And so the question is, can you build an encoding model that predicts and explains a lot of the variance in this fMRI activity? And so we leave the door open to do convolutional, recurrent, transformer, supervised, whatever you want. But one thing that we've been noticing, as I said previously in the talk, that we've seen this correlation between if the model is doing really well on the task, it's going to do really well for brain activity. But interestingly enough, we don't quite see that with transformers anymore. That starts to plateau. And it is this really interesting negative example of this. So maybe there's something there. Right now, the dominating models that are most brain-like are ResNets, essentially. So recurrent convolutional networks. So it's interesting to see if people can do any better. And so the challenge is live for a prize until July 26th, but it will go on indefinitely for you to continue like benchmarking and testing your model. Oops. So this is one of the data sets that we collected for that 2021 edition called Bold Moments data set. And so the idea behind this is that all the stimuli came from a subset of the moments in time data set, which is about 1 million short videos for action recognition, often using computer vision. And so we had 10 subjects viewing 1,102 of these short videos multiple times. We annotated them quite a bit with pretty high quality sentence descriptions, action labels, memorability scores, and so forth. So the idea behind this is that we're trying to advance our understanding of the visual system to go beyond just like these black and white sinusoidal waves, beyond the grayscale images, beyond still images, and now getting to short videos, because humans have evolved in this dynamic world that's constantly moving and changing. And other studies show that our brain is much more activated in response to these little short movies or long movies. I'm just more activated in response to motion than still images. So we want to try to model that. And so this is a data set paper initially. It's under review right now, but we do show some initial analyses to kind of get people thinking about how they might be able to use it in their work. And one of the ways is, again, we have so much more neural activity to work with, especially neural activity that goes into the dorsal stream and beyond the parietal cortex into action recognition networks. So some pretty cool stuff. And so we have analysis that demonstrates using a temporal shift module with a resonant 50 backbone that there is this hierarchy that goes on that extends into the dorsal stream and beyond. So this extends work that already showed a hierarchy through the ventral visual stream. Where we're trying to advertise that there's a lot more stuff to play with here. And so in this analysis, we took predictions from block 4, subtracted block 1 to see that hierarchy. And we also use this temporal shift module to then shuffle the frame order to see if the temporal matter or temporal order of the videos even matters, and turns out it does. So we do have temporally meaningful signal. The responses aren't just some smushed average of the frames. The brain does care about the order. And the last one I'll talk about, we do more in the paper, but we link the perception of these videos to another high-level cognitive function like memory. So I already touched on image memorability before. This is video memorability, exact same concept, but here we see that neural correlates of this are distributed throughout regions in the brain that are not seen with images. So this suggests that there's this deep connection between regions responsible for perception of the stimuli with then encoding it into memory. All right, last I want to touch on the future of DNNs in computational neuroscience, what that might look like, but this talk would not be complete without some fair criticisms of DNNs as models of the brain. So first one is the adversarial examples. So the argument goes, you take this model and you just add some imperceptible noise to it, and the classification is wildly different. Well, obviously the human brain, the human visual system doesn't work this way. So this can't be a good model of the visual system. Another line of thought is this idea of prediction versus explanation, where they say that predictive power isn't super useful. What we really want is an explanation of the visual system. And the analogy here is that an analog clock is a really good predictor of a digital clock, near perfect, but the internal mechanisms are completely different. So just prediction accuracy alone isn't very useful. It's like a call to kind of go back to the neuroscience inspired models. Another thing is the training diet for networks and humans is vastly different. No matter how you train a network, it's not going to look like what you see on the right, which is a baby neural network training by putting stuff in its mouth, crawling around on the floor. It's very multisensory, tactile, so on and so forth. And lastly, there are some biological implausibilities with neural networks. Like it's still unknown whether the brain can use gradient descent, for example, to learn. And Bowers et al. 2022. This is a bioarchive paper that is like a nice. It's a nice succinct attack on DNNs' brain models. So worth a read. But I and a lot of other people still have good reasons to be excited. It's hard to ignore the really good prediction power of these. Again, we've tried these neuroscience-inspired models. They don't quite work. And these DNNs do super well, even as you get to more and more complex stimuli. Even if you start to perturb it, translate it, rotate it, so on and so forth. And another advantage is that they're more interpretable than brains, at least. Every day they're getting more interpretable. So you can break open the DNN and see what's actually happening inside. So like that Murphy et al. Study did, I talked about at the beginning, you get this really good predictor of brain activity that represents a hypothesis about brain, about the visual system. Breaking into this neural network and investigating it is really valuable. And lastly, but not exhaustively, again, transformers do represent this really interesting mark of where task performance and brain prediction accuracy are looking like they diverge a bit. And that's very interesting because there's other lines of work that are, I went to a talk about it, so I'm not sure if it's published yet, but the gist of it is that they're looking more at the fundamentals of the CNNs and transformers and how they're fundamentally different from the brain in terms of like representative basis functions. And so some cool work there showing that there are indeed differences, and maybe CNNs are actually truly more brain-like than transformers, despite a transformer's better task accuracy. But nevertheless, this is an exciting new era to work with neuroscience and DNNs. So some things to look out for in the future that are on the horizon. This is actually already happening now, but training DNNs directly on brain data as these data sets become just bigger and bigger and bigger. That's a really powerful approach. And so instead of just saying, well, this is a jet ski, we can start to actually say this is the neural activity for this image. And so this idea of there's foundation models in computer science. Now we're starting to think of foundation models or brain foundation models, these models trained on brain data or trained to predict brain data. And so the community is looking for like Hugging Face type interfaces to be transparent and reproducible and to reliably use these models. And especially with GPT-4, people are starting to think, if not now, then soon, AI could just be another form of intelligence to study in line with everything else we study, like rats and bunnies and monkeys. And multimodal models are exciting as well, because now we have these networks that now start to predict simultaneously MEG, fMRI, behavior, ECOG, anything else for a more complete and unified account of visual perception. BCI interfaces can become a lot better, pushing to the next generation. Again, that Stable Diffusion paper is fun, exciting, something to look forward to. And then lastly, starting to think about how can we push these models beyond just looking at visual image statistics or language semantic statistics and into things that the models don't quite have yet. So like a memory mechanism or morality judgments or something that goes beyond that, well, I'd say at the 150 millisecond mark, like an object identification. So I just want to say thank you to everyone. These are my collaborators I work with a lot. So a lot of this work is supported by them. And also, again, thank you to Voxel 51, Jimmy and Jacob for having me.